Good morning, ladies and gentlemen. Welcome to the E-THERAPEUTICS PLC investor presentation. Throughout this recorded presentation, investors will be in listen-only mode. Questions are encouraged. They can be submitted at any time using the Q&A tab situated on the right-hand corner of your screen. Just simply type in your questions at any time and press send. The company may not be in a position to answer every question received during the meeting itself. However, the company will review all questions submitted today and will publish those responses where it's appropriate to do so on the Investor Meet Company website. Before we begin, we'd like to submit the following poll, and if you could give that your kind attention, I'm sure the company would be most grateful. And I'd now like to hand over to CEO Ali Mortazavi. Good morning.
Good morning, Mark, and thank you for having me on.
The floor is yours, Ali. Thank you.
Great. Okay, thank you very much, everybody, for joining. I joined E-Therapeutics 1 week before lockdown, which was a bit of an unfortunate timing, but really I joined after many years in the drug development industry. As you know, drug development and biotechnology is a kind of very long, very costly, very risky industry. And really my mission was, can we compute the future of medicine? Can we change the cost and time dynamics of drug discovery? Is drug discovery potentially a data science problem? And this has been really a journey of stitching up components by component, every piece of the process to really ultimately try and do as little wet lab and clinical trials as possible and to do as much computation as possible. Here's the kind of disclaimers, which I'm sure you've read many, many times before. Okay, so the starting point was really to start with a drug platform, and ours is an RNA drug platform, what I call our kind of picks and shovels, and we've branded something called Galonic. And really the starting point for me is that RNA drugs are information molecules. That's the way my representation of them is in terms of codes. Even though it's chemistry, I look at them as codes. And what do I mean by that? Is you can sort of see over here, this is a sort of template of our drug. You can see that over here, familiar genetic sequences to you. And really our drug is made of genetic sequences and how they interact with other genetic sequences. A very, very important, really long-term problem in the RNA world is RNA information molecules need to get inside of cells. That's a kind of evolutionary no-no, if you like, and the only thing that can get into cells successfully are viruses. Nevertheless, in a previous role at Silence Therapeutics and now at E-Therapeutics, we developed a drug delivery system. I won't go into the details of that, but in short, we attached the delivery system to the drug. And an important point about the delivery system is that it will take our drug to one cell type. So there are around 300 cell types in the human body. Our drug delivery system takes them to one cell type called hepatocytes, which will be an important kind of, if you like, data structure for us going forward. So to go through the steps of how information and information molecules work, the starting point is we know the code for every gene in hepatocytes, every single genetic sequence, we can look them up. So the starting point is if we suspect that this gene is causal in a disease, what we then do is leverage on another piece of computation, which is we create what's called the opposite code to that gene. So what's kind of relevant about the opposite code is that once inside of a cell, the opposite code, the laws of genetics say they kind of opposites attract and they bind to each other, which I'll show you on the left-hand side in a minute. So kind of, as I said, step 3 is really delivering this RNA drug to hepatocytes. What you can see is we give an injection and this is the wrong way around, and the circulatory system, if you like, takes the drug and the delivery system to the, if you like, the surface of hepatocytes, and there's what we call a sort of docking station. They kind of match each other, and then the drug is internalized. And if you like, at that point, our job in terms of the actual drug is done. The rest is done by the cell, by the process of opposites attract. The cell then basically finds the gene that we're interested in. You can see the process here where the opposites are attracting and binding to each other. And there's a form of, if you like, computation in a cell, which says when this binding process occurs, it will cut up, or if you like, silence the gene that's in question. So if we go back to the starting point, if the gene that we've chosen is causal in disease, and we can deliver the anticode into the cell itself via its own form of an algorithm, if you like, says that as soon as the opposites attract, as it does here, this gene's function will be stopped. If that gene causes disease, you hope that the disease process will be stopped. So if you like, that's a first layer of computation in terms of our drug. Going back to is drug discovery a data science problem? I would say this is a kind of important tick in the box. Okay, so this is an industry my team and I have had a long track record in, maybe more than 10 years of experience trying to do this. So we can sort of come up with some generalizable properties with our drug platform. The starting point is we can generally say that we can stop the function or inhibit the function of any gene in hepatocytes by the same process of looking up the code for any gene and creating the anticode. Kind of more importantly now is that this drug platform is now highly validated drug platform. There are 5 approved drugs. Tens of thousands of human safety and efficacy have gone through. So this is not an experimental system. This is, to my mind, maybe the most validated drug platform that there is. And here on the kind of right-hand side is what we can sort of typically expect in terms of almost the performance of the drug in every instance. And in simple terms, you can see here on the y-axis is number 1 is, if you like, whether a gene is fully functional. What you see here is a little bit blurred here is 1 injection on day 0. And the red line, as you can see, sort of coming down day 14, day 28, you can see that day 28, this arrow here, is showing that this gene's function is now reduced from 1 to 0.1 and around 90%. In fact, what's kind of important is that even on day 84, this gene's function is only 50% functioning. So we can generally say that any gene in hepatocytes with 1 injection and of the anticode, we will typically get 3 months of activity, and we will probably see around day 28 almost all the function of that gene, if you like, inhibited or stopped. So this has been quite a difficult process to come up with our own platform. RNA drugs are a highly protected field. We now have 17 patent applications to file, which we filed to protect our sort of, if you like, designs. But a very, very important part and last property which I want to sort of point out is this, is that from any gene selection in hepatocytes, we can make the drug, the final drug, for £500,000 in around 6 months. And just to give you an idea of how that compares to, let's say, another drug modality like small molecules, it can take up to 5 years, maybe even more, 7 years, and many millions of dollars just to make the drug. So already 6 months is not quite what I call computation, but still it's extremely fast. We're dealing with information molecules. Comparison to the current drug discovery world out there, £500,000 is extremely, extremely cheap, if you like. What is the value of a kind of drug platform, a therapeutic modality? This is an example of what are called monoclonal antibodies. Quite unlike iPhones, you can see that a drug platform like monoclonal antibodies started in 1975 is still going very strong even in 2023. They're very rare to come around. They're very rare that a drug platform comes around very often. If you want to have a comparison here, the red arrow pointing up, I've got lots of red arrows here. We're in around 2002 in the monoclonal antibody world. A very, very long way to go in terms of sort of opportunity space. Okay, so the picks and shovels, they work. They, as you can see, are a form of computation, information molecules, and leveraging the cell's own algorithms. So the first part of the question, I think, for me is kind of answered, that yes, that aspect can be a data science problem. So now the question is, okay, but which gene in which disease within hepatocytes are important? And the field of molecular biology and gene targets and diseases is, I would say, extremely primitive, and maybe one of the most underknown sciences compared to kind of physics and chemistry. So to us, though, given that we know that our drug always goes to hepatocytes, we are interested in what we call hepatocyte biology. There's a sort of, if you like, zooming in process to that one cell type. We're only interested in that cell type. One point I would make is that as hepatocytes make more than 90% of the liver, really the organ where we go to is the liver. There's a kind of interesting role play in the liver, is that the liver is perhaps the most active organ in the human body, really what I call the central trafficking system. 12% of our blood is bathed in the liver. The liver processes all the nutrients, all the toxins. So the liver is, if you like, a central processing unit where things come in. And as you can see, the liver actually, or hepatocyte biology, can really have outcomes in what I call very distant organs. You know, kind of counterintuitive to think that a gene in the liver could have an effect in an eye disease, which is at least theoretically distant from the liver, if you like, or brain diseases, or in both. And that's because of the function of the kind of, if you like, this central CPU unit. So in terms of, if you like, going back to sort of computation and data science, these are just some of the sort of high-level numbers that we're interested in. There are around 12,000 genes in the liver, around what we call 1,000 secreted proteins. What do we mean by that? Is that the liver processes proteins, let's say cleanses them, processes them, and then circulates them via the circulatory system to these distant organs. There are also what we call 700 biological processes within hepatocytes, and I'll come on the next slide as what I mean by biological processes. But an interesting point for me is that we're not a liver company. Like, for example, we are not solely interested in liver diseases. And that, again, is because of the kind of central trafficking role of things coming in and being distributed out in the liver. So, if you like, genes expressed in hepatocytes affect cardiovascular disease, diabetes, or blood disorders, obesity, kidney diseases. So we have this kind of strange position where we've really zoomed in into, let's say, 1 out of the 300 cell types in the human body. Our actual addressable market size is extremely large because of the kind of liver's role in the human body. Computationally, that's what's interesting for us, is again that zooming in process, which basically means that we can now only be interested in all the data sets which are sort of, if you like, active in hepatocyte biology. So what we see in our industry is these are kind of some of our competitors. Again, what this is showing is RNA drug platforms are very, very rare. Very, very few people work in this industry. One of our competitors, Dicerna, got taken over by Novo Nordisk around November 21 for 3.3 billion. But my point here really is these are the genes in the liver which these companies are working on. And what you'll notice, there's a high level of congestion, if you like, 4 companies working on, let's say, this gene, Lp(a). And for me, what I saw is a very, very large opportunity space that, you know, there are so many genes and biological diseases in the liver. It seems to me inconceivable that the universe should be so tight and narrow. And again, I wanted to apply computation to that. And I think I want to show you how I think we've been successful in finding gene targets and disease sort of pairs that are not really on anybody else's pipelines. Okay, so the next stage for me is we have a very important function, and we are able to model biology in a computer. So going back to biological processes, remember I said there are 700 biological processes within the liver. One of them is the clotting cascade, literally clotting. So how do we go about modeling, let's say, the clotting cascade? Is by generating these kind of weird and wonderful looking networks of genes. So what you see in the nodes here are genes which are interconnected with each other, forming a very kind of robust biological system. If there's something going wrong in this kind of biological process, what we do is we can design these biological processes in a computer. We can analyze them in a computer. And more importantly, we can find what are called the best target genes, the hidden players, if you like. And what you can see is in this particular example, you would think that intuition should tell you that the most important gene in this network could be the big circles of the ones in the middle. But actually, usually in all networks and biological networks in particular, the really important players are kind of hidden. So, we can basically analyze and find these. The idea being that if you can basically use our drug platform to inhibit this particular gene, this biological process will get disrupted, and you can improve, let's say, the clotting cascade. So, we have the ability to model every single biological process in hepatocytes, computationally. So where does that kind of lead to is, as I said, hepatocytes and hepatocyte biology is our primary interest. So we have developed what we call HapNet. HapNet is really a central data resource, what we used to call a Google for hepatocytes, and I'll come back to this phrase, Google for hepatocytes. What does HapNet consist of? First and foremost is what we call unstructured data. What do I mean by unstructured data? Well, take a scientific paper. A scientific paper is usually text, graphics, tables, sometimes even videos. But in essence, we have imported, downloaded, have access to every single hepatocyte biology related paper, grants, proteins, all the competitor drugs, pathways, you name it. We probably have the largest biological resource for hepatocytes. And on top of that, we have our, as I said, ability to model biology. HapNet is, if you like, our GUI. Then what happens is, going back to, we want to do as much computation as possible. The problem we had is that really comes back to the Google of hepatocytes. If there's in effect way too much to read and understand, let's take a paper in the clotting cascade. We've probably got about, let's say, 5,000 papers in the clotting cascade. At some stage, our sort of team of competitive intelligence, which are medics, chemists, biologists, and people can sort of model the financials of a particular disease. You go from doing things extremely fast. If you like, you're able to search, you're able to refine everything to do with hepatocytes, then let's say clotting, then papers. But at some stage, you hit the slow lane where somebody and a team has to read and assess what the outputs are. And maybe this isn't just the fault of E-Therapeutics. Going back maybe 6 months, we all, irrespective of whether we're in hepatocyte biology, but if we want to research a company, we would go to Google. Google was great in terms of some sort of search function, but it would generate a list of possible, let's say, websites or resources to go to over many pages, and then began the manual process of going in, looking, saving, etc., etc. So very similar to kind of this process. Nevertheless, what I would say is the combination of, if you like, that resource of HapNet, modeling biology, hepatocyte-centric data, experts in hepatocyte biology and chemistry. We have, as I said, filed 17 patents. We have 6 novel gene targets, which we're prosecuting, what we say in our preclinical process. There are in 5 therapeutic areas. What you can see over here is we have these targets which are looking at diseases in hematology, blood disorders, cardiovascular disease, NASH and fatty liver, obesity, and a kind of, again, strange one here, ophthalmology. Remember, a particular gene and stopping its function in the liver could have a distant outcome in the eye. And we're active in these. They're going through our preclinical cascade, and we hope to see success with some of them this year to be able to go further. Okay, so really the main thrust of this presentation was about generative AI and how that's changed what I think is, let's go with the phrase Google of hepatocytes, to something very, very different. In actual fact, we've been, let's say, experimenting with generative AI models probably, since last year, actually. But for a long time, we were disappointed with some of the outputs. We could see the potential in them, but we didn't really see how they could have an immediate business impact. We have obviously data scientists, machine learning programmers, computational biologists, but they were very much kind of in the background. But around, believe it or not, March this year, something very, very dramatic happened. It completely swung the entire computational strategy and possibilities of this company. So what happened? I've highlighted the number 10 to the power of 22 in each of these graphs, which I think I'm going to come back to. And in simple terms, let's start with what is a large language model? What are these generative kind of models? In some ways, they're incredibly simple. They really just predict the next word, which you would think as a sort of basic function shouldn't arise anything particularly special from it. But what I saw around March of this year, and really I think has completely changed the landscape, and maybe one of the reasons why all of a sudden everybody is talking about doing this in different kind of industries, is there seemed to be that the combination of predicting the next word and what's called the size of the neural network. At a certain size of neural network, the function, the actual output of these systems dramatically improve. And one of my favorite examples is Persian Q&A. So you imagine a system like GPT-4, it's not in any way trained on Persian, let alone Persian Q&A. But it seems that at a certain size of model, it is very proficient in not only, let's say, talking in Persian, but being able to converse in Persian. Maybe one of the best, if you like, descriptions of why this is happening is by the chief scientist of OpenAI. The real answer is nobody knows exactly why this kind of size and function emergent property has happened. But as he kind of says here, predicting the next word derives an understanding of the reality described in the various materials. And I'll come to this word understanding because I've underlined it. I'll come to, obviously for us, Persian Q&A is not particularly useful in hepatocyte biology, but how this kind of translates for us into something very materially different. Okay, so, part of being a small company is you suffer a lot from being small, but one of the advantages of being a very small company is you can walk in the next week on a Monday and say, okay, we're putting generative AI at the center of absolutely everything that we do, HapNet, if you like. Everything that we do has a generative AI core. Kind of everything has been, if you like, switched around to incorporate the capabilities of generative AI. So, an important point for me, and obviously this is quite a complicated subject, is for us, we're not using a chatbot. So I'm sure a lot of you have used ChatGPT, which is an incredibly powerful, if you like, capability. But I think it's kind of missing the point of what is really possible and the power of what's really possible under the bonnet and what we kind of use it for with our data science and machine learning programming team and computational biology team. So for us, a few things that are very important is we can train our own specialist agents. So what do I mean by that is if you take GPT-4 or ChatGPT, it's, if you like, an expert in absolutely everything, but it could talk about, well, hepatocyte biology up to a point. It can talk about glasses of water. It can talk about physics and everything. It's almost too broad. But for us, what is really exciting is the capability of feeding a generative AI model, let's say hepatocyte-specific data, so it becomes almost an expert in that particular field of, let's call it, hepatocyte data. And I'll give some use cases which will sort of really, embellish what I'm kind of talking about. So we fine-tune individual, if you like, agents or models, and really what's surprisingly profound, really, is none of these things which were possible. So go back to, we were also limited by the Google of hepatocytes, but now what we call our specialist agents can, and I put them in speech marks because can systems really understand, reason, or infer? Some of them are questions of philosophy, but yes, they can. So for example, training a model on hepatocyte-specific data can understand all of that data in one point. Just simple scale, we can import 10,000 papers on a particular disease, let's say. It can ingest, it can learn, it can basically tell us exactly what's going on. These agents we can also code, but they also have an ability to self-code, which is quite a frightening process, that they can sort of self-iterate and improve. They have access to the internet, so kind of live data that's coming out. They have obviously access to our own internet. So to my mind, what was a sort of hazy picture of, let's say, computing the future of medicine is drug discovery, a data science problem. At this point, almost in March, was the missing component, the kind of almost slotting in, which really completely changed, in my mind, the fields of really biology, chemistry, and medicine. And if you like, more lucky than good, we were set up as a strange biotech company, because most biotech companies don't have machine learning programmers and data scientists. But we went with that mission statement of computing the future of medicine. We were just missing this kind of unknown quantity. Okay, so a couple of use cases. Hopefully we can sort of give a bit more detail on what the sort of ultimate outcome is for us. As a starting point, as I said, we have gone from Google of hepatocytes, search heavy, the human being being a very big component, even though we sort of really zoned in on hepatocyte biology and all the kind of modeling that we had, we were still reliant on a human element. With generative AI, we have now added an unbelievable amount of automation, ability to be able to ingest any single one of these, let's call them unstructured data. All the scientific papers we can import and read and understand and reason in a completely different way. The outcome being that I don't think we'll ever want to or can get rid of, if you like, the human element of it. But it's going to be a much, much smaller component. And at the very least, you know, at its core, what I can say is, if there are 10,000 genes, let's say, in the liver, before we would be processing them at a certain rate, and obviously humans have a slow rate, we are now capable of, if you like, processing every disease, every gene, all at the same time. And think about a step change there in terms of being able to do that, ingest, understand, reason, and then basically be able to come up with outcomes. So a kind of good use case for unstructured data is patents. This is a good sort of couple of pages on patents. You can see there's, you know, tables, bar graphs, it's chemical data. This is what we call unstructured data. It's difficult. Human being had to read patents. One patent can easily be 300 pages. We had really had a very important project, what we call the sort of old approach pre-language models. We imported all 400,000 RNA-related patents, but we had a serious limitation. And actually, this slide is a good example of what I call easy wins, and in terms of AI and what can become really very sort of complex and if you like, clever. So the first process is, in the old process, we got these sort of 400,000 patents. Could we have summarized and categorized all these patents? Not really, not without a lot of human intervention, maybe almost impossible. Now, as I showed you with our sort of ETX patent agent, that's very possible. If you want to go to a sort of, you know, let's say less easy wins, could we have previously understood, in inverted commas really should be, the syntax of patent documents, all 400,000 of them? That would be a manual process. Maybe nobody in the world has read and understood all these 400,000 patents. Is that possible now? Very much so. At its kind of ultimate form, could we have, you know, computationally auto-generated patents? Can it literally write patents itself? Absolutely not before. Absolutely yes now. So, you know, maybe the takeaway from this is the number was very large. You can imagine that any human intervention of reading, you know, sorting, inferring, categorizing was almost impossible. Completely changed in terms of our ability to do that. Patents have huge amounts of biological information, chemical information. Our ability to incorporate that into HapNet and be able to scalably understand and get new outcomes, really, to be honest, game changer. Okay, so this is a project which is really quite fascinating. So far, we sort of probably talked about language, words, if you like, ChatGPT, even our sort of back-end agents have been talking about words, predicting the next word. But actually, these AI models are not really just about words. They're about any sequence of, if you like, text. So a very important project for us is this is a particular gene that we're interested in. And you can see this is a sort of 2D structure of the gene in just raw text. This is the genetic sequence of it. In reality, if you go into a cell, the gene is a 3D view of the gene, kind of has this weird and wonderful structures. It doesn't really kind of look into a 2D shape, as you can see there. Sort of the third component, as you've seen before, is our sort of drug platform. So one question for me was, is there a secret language in genetic sequences that maybe these AI models can understand that we humans, not just E-therapeutics, can never understand? Ultimately, can we design and predict the activity of our drug entirely in a computer? And the answer is very much on the way to doing that with what we call our ETX RNAi agent. Is there a secret language in genetic sequences? Yes, there is. How does it work? Well, our first sort of, at the moment, training model, we have fed 13 million genes into this AI model. 13 million genes of every possible species, not just humans, that you can think of. And kind of, we took the same approach as, if you like, language. What you do is you mask out certain sections of a particular gene. So let's say if we were to sort of go here, we would mask this little section out. And we would say basically to the model has to predict what the missing sections are. And in the process, very similar to language, of predicting the next word or predicting the missing sequences, in that 13 million genes, which no human or any system could really look at in this way, yes, it has started to understand the representations of genes. What really that meant is understanding representations of genes means that we can then say to it, okay, where's the best RNA drug that we can basically come up with in a computer? So kind of what does this mean, as we stand is, we can predict of any gene that you give me or I find 6 of the best RNA, if you like, drugs that can exist. And that performance will rapidly improve. What does that mean for us is, again, let's go current approach or old approach, new approach. So previously, we would find a gene. We think this gene is causal in disease X. We would then find 400 anticodes, if you want to. And at some stage, we had to test those anticodes in a wet lab. That would take 6 months and £500,000. Pretty amazing compared, as I said, to the small molecules. What we're really looking at is to do the entire thing computationally, i.e. finding 10 in a computer, maybe even less, making it in a month, maybe even less, reducing the cost from £500,000 to £50,000. Even less is possible. So you can see that in every aspect of our sort of drug discovery, computation via generative models are starting to make profound differences which weren't possible before. Okay, so kind of what is the impact of this? It's got a few sort of very important points. Ultimately, I think this is an important point. There's a significant reduction in wet lab work and even clinical trials. Really, I think what's becoming is clear is computing the future of medicine is becoming a reality to the point where, let's say, a lot of the prediction and probabilities can be done computationally, and less wet lab work, less and less, even if you like, clinical trial work, so that the costs and times of this very expensive industry are going to have a material change. And for us, what we're really kind of developing is a generalizable model for drug discovery. What do I mean by that is our learnings, our systems in hepatocytes using this picks and shovels is really generalizable to any cell type. Or the tools that we're developing, for example, the RNA computational drug developer, we can try and develop to anywhere. And that kind of means for us is that, for me, that is drug discovery a data science problem? Yes. without any question. Is this industry now computable? Very much so. It's obviously a process and will take some time. But this is the significant, if you like, intelligent system piece that was missing. Okay.
That's great, Ali. Thank you very much indeed for updating investors. Ladies and gentlemen, please do continue to submit your questions. But just while Ali takes a few moments to review the questions submitted already, I'd just like to remind you that a recording of today's presentation, along with a copy of the slides and the published Q&A, can be accessed via your Investor Meet Company dashboard. Ali, you've had a number of questions throughout your presentation. So firstly, thank you to everybody for your engagement this morning. Ali, if I may just ask you to open up that Q&A tab, and if I could ask you just to read out the questions and then give a response where it's appropriate to do so, and then I'll pick up from you at the end, if I may.
Sure. I have a question here saying that given that AI large language models potentially offer so much to E-Therapeutics, does their occasional known tendency to hallucinate undermine their usefulness in areas of research that depend so much on accuracy? So two things I would say. I would say that, let's say, the fields of biology, chemistry, and medicine are, as I use the word, so primitive in that, not just at E-Therapeutics, but generally our understanding of human biology is so little that it doesn't take almost anything to change the probabilities of new knowledge, new findings, etc. That said, hallucinations are a problem, a real problem with these language models. We are developing systems which kind of self-reference, self-check. That probably is why you need a human element. But designing these agents also includes designing agents which are able to self-reference and self-check and be able to sort of, for example, one of the things we're working on is what we call a sort of truth database. That's just a simple thing where outputs can be looked up to our truth database, etc., etc. It is a problem, but I would say that in our field, the white space to aim at is so huge. It's, to my mind, at this moment, not as big a problem as the opportunity. So, second question from Christy. How advanced is E-Therapeutics in terms of AI compared to others in this space? I would say that in the field of, let's say, in the sector of biotechnology, so remember, Chris, we were set up to do this without necessarily knowing that this was going to come along. Set up meaning that it's, as I said, rare for a biotech company to have tech people, if you like, or even start with that question: can this be a data science problem? The field of drug discovery and drug development is very much empirical. You've got to do the experiments, you've got to kind of do that. And obviously we're a part of that, but we started with this, if you like, mission statement. In terms of, I think what's kind of, to my mind, interesting is haven't reinvented the wheel or our business model. And also, we're focused on one cell type, a very active cell type, and we have a very, very kind of unique and highly sought after kind of, if you like, picks and shovels. I think that's an important edge to have, certainly across the people, sort of our competitors in that field. We have a completely different skill set and mindset. But we have, been developing systems and algorithms and data sets for, well over 3.5 years now, which, obviously in time, I think generative AI models will take over all of pharmaceutical companies or whatever, but I think we have a very important edge on multiple levels. Do you want me to choose another question, Mark?
Yes, please, Ali, if you wouldn't mind, if there's a few there to pick the bones out of, if you'd be so kind.
Mike T, can you explain a little bit about our competitive barriers to entry given the generic availability of AI? So the first thing is availability of AI. So the first thing is I differentiate between ChatGPT and what we call AI. That what we call sort of AI and being able to use APIs, machine learning programmers, training models, is a very sort of specialized field. It's not that accessible. You have to have the expertise. Second of all, we have, let's say, the domain expertise we have in-house, hepatocyte biology experts, RNA chemists. So in our domain of interest, I think there's a very, very high barrier to entry. There was, even before generative models, for example, having the IP and the know-how in RNA interference is very, very rare. Having that kind of hepatocyte focus, even though I downplayed it as the Google of hepatocytes, is a very important resource. Again, time sort of stands still for no one, but I think we have a very major advantage in terms of a competitive advantage over competitors. I said competitors, too many there. I'm looking at another question. What regulation do you think is needed given the impact AI could have on medicine? So, to my mind, any system which understands, in inverted commas, and reasons and infers from human language, human knowledge. So remember, these systems haven't memorized. It's not like they've got an encyclopedia. They are inferring and reasoning. Sometimes they hallucinate, but in general, they're inferring and reasoning and understanding very well. So any system which can do that and have code, self-code, access to live internet, by definition, is a very powerful, maybe you can call it dangerous system. But in the fields that I'm interested in, biology, chemistry, and medicine, they can only really do good. If the question is, can these systems do harm with killer drones? Yes, it needs regulation, but not in the fields that can sort of improve human outcome. I'm looking at a question. What interest, Mark A is asking, are you seeing from larger pharma companies in terms of the impact you can have on drug discovery and the time to bring in new drugs to market? The answer to that question is, if you go back, let's call it a year, there's been if you like, a disconnect between computation and wet lab drug discovery and clinical trials. It's been hard to sort of, for partners and even for a company like us, let's call it the Google of hepatocytes, is that human intervention in the middle meant that you had to go and do things in the wet lab much more than, let's say, even we wanted to. So what we were coming across is, okay, we'll go and prove it in a wet lab, in a sort of animal model or a clinical trial. What I'm seeing in the last 6 months is a complete shift in interest from the wet lab to what have you got computationally. And that is because I think people are really beginning to understand this word, understand, infer, ultimately come up with probabilities, if you like. So I would say the balance has massively changed, and the phrase AI drug discovery, which was really a non-sector in some ways because it was in no man's land, has completely changed in the last 3 to 6 months. When do you think you will significantly commercialize this opportunity, and do you have sufficient funding in place to get to being profitable? I mean, those are kind of difficult questions to answer. To my mind, what I'm seeing here is something very profoundly different, which I personally didn't expect. We kind of sensed that it was missing. At the moment, I'm just very, very focused on the opportunity space, capitalizing on our sort of lead, on being able to sort of add resource to it. Do we have sufficient funding? We can calibrate the funding to as little or as much as possible. To my mind, from what I'm seeing, this is a time to, let's say, put the foot on the accelerator, because maybe to me, the most profound technology I've seen, I hate to say it, but probably bigger than the kind of internet.com boom, if you like, by a big number, because it's usable. Might have. I mean, most of the kind of questions that I'm seeing here are all about kind of partnerships and that kind of business model. So We're still the same, if you like, drug discovery company. We will still have to do some wet lab work. I don't think we're at a stage, or anyone is at a stage, who can do everything in a computer, hallucinations being one of the kind of reasons. But if, let's say, computation was, let's give it an arbitrary number, 20% of the process and 80% of the process was wet labs and clinical trials to validate, I'm now, in my opinion, seeing a swing the other way, where the wet lab and the clinical trials become a much smaller component and the computation becomes a much bigger part of the probability kind of process. One question here is, have they approached ETX to use your database, or are we still too early to see ETX monetize? We have lots of discussions with so many different people. I would say that, would I like to partner something? To my mind, what I'm seeing is, I think we're going to see a world where large pharmaceutical companies will do what they're really good at: clinical trials, scalable lab work, etc. And maybe smaller companies like E-Therapeutics can be what they're good at, which is, being very fast, nimble teams, being able to do things like come over the weekend and say we're doing generative AI, end of story, rather than having 2-year meetings about hallucinations or, the kind of security processes, which are important. But this is the time that, in my mind, multi-billion-dollar companies are going to come out of nowhere. And that's what I'm really focused on, is to really capture that opportunity.
Ali, as I say, I think you've taken a lot of questions that seem to be around the same thing, around partnerships and things. I think thank you to everybody for those questions. I think you've touched on those. I don't know if there's anything further there, Ali, that you can see, or whether or not, if I may, as I know investor feedback is important to you, I'll redirect those on the call to give you their thoughts and expectations. But if there aren't any further questions, then maybe I could ask you just for a few closing comments, just to wrap up with. And then, as I say, I'll ask investors to give you their feedback.
Yeah, I mean, my closing comments really go back to the first slide here. I think this two kind of mission statements and questions have unquestionably become a yes and a yes. The second question for me is, how do we best capitalize on this? One of the things I think we've got to sort of all be aware of is, literally every day or every week, there's new language models, there's new what's called transformer models, there's new processes, new products, new software coming out. And some other hesitation in, well, what is the exact business model, is basically the same, but there are things that are happening. For example, the ability to be able to look at what we call the secret language of genes, which are completely unknown in this industry even a few months ago. We have to be careful not to do too much, but at the same time, we have to be careful not to be conservative.
Thanks ever so much indeed. Could I please ask investors not to close this session, as we'll now automatically redirect you for the opportunity to provide your feedback in order that the company can better understand your views and expectations. This won't take a few moments to complete, but I'm sure will be greatly valued by the company. On behalf of the management team of E-Therapeutics PLC, we'd like to thank you for attending today's presentation, and good afternoon to you all.